Welcome everyone. I'm Agnese Wimpy from NanoString Marketing, and it is my pleasure to open the second day of the Spatial Answer Trilogy. Yesterday we focused on oncology, today on immunology, and tomorrow on neuroscience research. Please note that this is a live webinar, and we encourage you to ask your questions using the question and answer box you find on the left of your screen. We will answer all of your questions if we can today, live, after the presentation. And now it's my pleasure to introduce to you our chair today, Dr. Doug Brown. Doug is the Chief Executive of the British Society for Immunology, the BSI, and leads its mission to support and transform immunology research to the benefit of human and animal health. Using the global reach of the BSI, they're building a best-in-class membership offer aimed at supporting research and immunologists at all stages of their career, as well as leading national and international initiatives to create a thriving environment, a thriving immunology research sector, and ensure that research is translated into the clinic. Doug has an immunology background, having completed a PhD at Cambridge University on HIV research, the postdoctoral position on malaria and stem cells. Doug worked for the MS Society, leading their biomedical research program, worked on the Alzheimer Society as Chief Policy and Research Officer, and he led a number of high-profile initiatives, including the very important one, establishing the 300 million UK Dementia Research Institute. Doug is also trustee of the Association of Medical Research Charities, and it's really a pleasure to have him today chairing the session. Doug, over to you now.
Thank you, Agnes, and I must say the pleasure is all mine. So thank you for that kind introduction and welcome everybody to this afternoon's session. I don't know how many of you know the British Society for Immunology or are members. I would hope a lot of you are members, given that you're tuning into an immunology webinar. But if you're not, please do visit our website, because there's a lot that we do offer our members, access to events, to an annual congress, career support, travel awards. We do lots of advocacy on behalf of immunology, influencing policy and the government, and work on a number of exciting initiatives across cancer immunology, vaccination, and lots of other areas. So I guarantee you, if you're not currently a member, become a member, you will not be disappointed. And one of those exciting initiatives that we have been working on over the last few months is, in fact, this partnership with NanoString and Illumina, which has been a fantastic partnership. They've been a great couple of companies to work with to bring this award and this technology, cutting-edge technology, to two award winners for free to support immunology research. And so it's been a real pleasure, and we will hear from those award winners later on. I will come to those, but before you hear from the award winners, I want to actually hand back to Agnes, and then Agnes will hand on to another colleague at Illumina to give some presentations from the companies. So Agnes will speak first from NanoString. Agnes, over to you.
Thank you very much, Doug. And I just would like to have to give you an introduction on the spatial biology that we're going to cover today, just to give you a background. So understanding the organization of cells and tissues and how this organization influences the function is a fundamental question in life science research. But why is this the case? So you can see in this schematic here an example of this point of the why. When adopting a bulk RNA analysis on maybe responders versus non-responders, for example, you might get the same results as indicated here in the heat map. When you instead interrogate specific areas in the tissues, maybe an immune-rich area versus an immune-poor area, you can really see the difference that was averaged out in the bulk analysis. So going spatial is very important to find biomarkers. So basically here you see the geomics where basically we merge the power of high-plex gene expression and the resolutions of imaging. And so with geomics, you can really resolve in situ the full transcriptome and understand complex tissues and cell interactions. So the flexibility to apply spatial genomics on sort of hypothesis-driven or discovery-focused experiment has been really noted last year. And in fact, in 2020, the Nature Method of the Year was indeed the resolution of the Transcriptome Institute. And another thing, we are very excited and honored that genomics was selected for such important journal and paper and accolade, in essence. So let me just, talking about paper, let me just show you some of the ones that have been already published with genomics. We have about 50, they're all peer review, of course. Some made the cover in important journals like Nature Medicine, Cancer Research, Nature, and you can see that your peers have been really active already in the field of spatial biology. I want to tell you a couple of words about the technology so that you can sort of follow a bit even more the presentations that are following after myself. So the technology is really based on either antibodies, if you want to measure proteins, or if you want to measure RNA, you use in situ hybridization probes. But for both cases in our technology, we leverage a special DSP probes that are essentially indexing oligos that are attached to the molecule they are attached to via a photocleavable linker. So the DSP barcode has a unique sequence, which is really unique to the molecule that it is attached to. And also, as we said, there is a photocleavable linker identified with the dot here, which is really release the DSP barcode when UV light is shown. Now, the workflow itself starts with the staining of your slide. So basically, the step one is staining and hybridize a cocktail of either in situ hybridization probes or antibodies labeled with the DSP barcode to your tissue, together with morphology markers that are fluorescently labeled so that you can see your tissue. And in step two, select your region of interest. In the picture here, the region of interest is represented with those circles, but you can really follow the morphology of the tissue and decide that you want to investigate a specific part of your tissue. For example, an example in immunology could be that you want to measure the RNA contained in the part of the tissue that has been infected by COVID versus the part of the tissue that has not been infected by COVID. So you can instruct genomics to shine the light, the UV light, on those specific areas to release the barcodes on the region of interest that you are interested in. These barcodes are then collected and dispensed on the 9612 plates that you see there, and then eventually counted either on the encounter or on one of the fantastic Illumina instrument next generation sequencers. And this is why we have a great collaboration with Illumina, because our workflow is absolutely connected. So once the barcode is counted, you have a file represented here in the table. That table is then put back into the software in the genomics instrument that really puts back the information, the high-plex gene expression information back into the tissue. So with this, I hope I've explained and shared with you, and you can appreciate how genomics can help you detect all targets, measure proteins as well as RNA, explore any region that you would like to investigate on SFP tissue, archived tissue, as well as fresh frozen. And of course, it gives you the flexibility of profiling large compartments or even specific cell types. So, and I look forward to listen to the presenters later on, which will make this all very, very real with real biology and data. But for now, I pass it over to Pablo.
Thank you very much, Agnese, for this great introduction. And thank you very much, Doug, for chairing this session. So my name is Pavel Sayak. I'm part of Illumina's marketing team, and I have the great pleasure of, in the next 2 or 3 minutes, to briefly present who we are and what we do. And I really find this field of spatial biology so exciting because biological systems, tissues, organs, they have a precise organization because it's important to their function. And being able to look at expression within this tissue context is truly groundbreaking and can tell you so much more about biology. And Illumina, we are a global company, and we develop, manufacture, and market innovative next-generation sequencing and microarray technologies. And our technology enables you to make foundational discoveries, to transform medicine, and ultimately to impact lives. And to date, there are more than 200,000 publications where our technology has been used, so it clearly shows that this is a robust technology, giving you results that you can trust. And you can either purchase a system, implement it in-house in your own lab, but you can also get access to our technology through one of the many service providers that are found throughout the world. And we do offer next-generation sequencing. And depending on how you prepare your sample for next-generation sequencing, so depending on how you do the library preparation, this opens up a wide menu of biological modalities. So you can study different facets of DNA, you can study different types of RNA, you can do work at the single cell level, and as Agnesa very nicely illustrated, you can also do spatially resolved analysis. And our sequencing system, in particular the NextSeq 2000, which you can see here giving you 1.1 billion reads, and the NovaSeq system giving 20 billion reads, they are an excellent match to the nanostring geomics assays. So the Cancer Transcriptome Atlas and the Whole Transcriptome Atlas, giving you the power to do those discovery studies to find out more about your biology. And we definitely also want to streamline the data analysis part. So now you can find the geomics pipeline in our base-based Sequence Hub, which is our cloud-based solution for data analysis and data storage. And this particular pipeline will basically take the reads, and it will create a digital count conversion file. And you can then upload that file into the geomics digital spatial profiling to do analysis and to do visualization. And if you have any questions about the sequencing solutions, we are more than happy to assist. So please don't hesitate to get in touch. So with this, I wanted to very much thank you for your attention and hand the word back to Doug.
That's fantastic. Thank you very much, Pavel and Agnes, for your presentations. Great to see these technologies and how they can be applied. And now, ladies and gentlemen, it's on to our award winners who have and are planning to use these technologies. Now, I saw the award nomination process when it was happening a few months ago, with applications coming in across the whole of Europe. There was fierce competition for these awards, and the judges reviewed them all and decided on 2 award winners, and have noted that the award winners were presenting innovative research and really exciting ideas that did deserve accessing this technology for free to further their research. So the award winners are, as you've probably seen already, Professor Susanne Krasemann and Dr. Michelle Naughton. Today we're going to hear from both of them, and then after that session we will have a Q&A session. So both speakers will speak, and then I will ask questions of each of the speakers. So please do get your questions in. There's a question submission functionality on the webinar platform. Get those questions in, and I'll ask as many of those questions as I can of our speakers. So welcome, Susanne. And by introduction, Susanne, as I'm sure many of you know, has carried out her research in various academic centres across the world. Susanne has previously worked at the Harvard Medical School on microglia dysfunction and prion clearance, and has worked in the virology and immunology research fields in Göttingen. Today, Susanne is the group leader and head of the core facility of mouse pathology at the University Medical Center Hamburg Eppendorf Institute of Neuropathology. And today, Susanne will be presenting her work on the blood-brain barrier dysregulation in COVID-19. Susanne, over to you.
Oh, Doug, thank you very much for this nice introduction. I'm very excited to be here. And as Doug already pointed out, I'm really sitting in the center between immunology, brain diseases, neurodegenerative diseases, and high-quality immunohistochemistry. And today I would like to present you our work about blood-brain barrier dysregulation in COVID-19. And although COVID is a respiratory disease, it's becoming more and more obvious that there are also severe like side effects on the brain. So you have neurologic dysfunction in severe COVID cases, but also in survivors and patients that recovered from severe COVID. You have the so-called long COVID, which is also going hand in hand with the neurologic dysfunctions like dizziness, headache, and but also more like severe things like seizures and stroke. So we were really interested what's going on in the brain. And the city of Hamburg is also a state within Germany, so it's a bit like independent. And when COVID started in March last year, the authorities of Hamburg ordered every patient to be autopsied to get like a hand on this pandemic and also on the underlying comorbidities to find out more about what is happening in severe COVID-19. And from this, we actually investigated more than 40 cases that died in the first German wave of coronavirus disease. And from this, we actually checked the brains to see what is going on. And actually, there's not too much going on because we checked for markers of astrogliosis, GFAP, in like microglia activation, invasion of peripheral cells like T cells here in CD8. And if you see the overview, you see that it's quite chaotic. So this is what I actually wanted also to show you, that there's not really a trend. So there are patients that like present with astrogliosis, but there are also patients that do not present with astrogliosis. So it's a rather fuzzy picture that we are seeing here. And obviously we were interested in the fact, is SARS really entering the brain? And if so, where is it going then? And for this, we first sought to establish a really decent immunohistochemical staining to detect SARS CoV-2 infected cells. And to do so, we first infected cells and poured them into paraffin blocks because obviously for patient material, the tissue of choice is always like embedded paraffin blocks. You because of biosafety reasons, you rather never work with fresh tissue. And a lot of people might know that this is always a bit tricky to work with paraffin embedded tissues. Thus, we checked this first. And we infected two types of cells, so virus cells, which are like a very great replicator for a lot of human pathogens, and human neuroblastoma cell line, the SHSY5Y cells. And then we did not infect here with the red arrow and tried a lot of different antibodies to see if they work on paraffin embedded tissues. And as you might see already, there are antibodies that do not work at all. Then there are antibodies that show like a lot of background and are thus not suitable for like detection of SARS-CoV-2 in paraffin sections. But then there are also antibodies that work really nicely. So with this, we moved on to like tissues, and these are especially good ones. So we first checked in lung tissue, and then we could see, wow, yes, it's working very decently in lung tissues. It doesn't give you any background, but then recognizes very nicely and very specifically cells that are infected in people that are like deceased with COVID-19. And but I have to tell that these are all lungs of people that died from COVID-19. So it's not that you find it everywhere. This was also something we had to learn during our study. It can look like this, but there can also be just single cells being positive like this. And there are also a lot of even lung tissues that are not positive at all. So going back to the cells, I mean, virus cells are very efficiently infected and titers rise very easily. And in the neuroblastoma cell line, it was not as strong as it was in the other cells. So the scale is much smaller here. You see that there is some infection ongoing, but it's the degree is much smaller. So neuronal cells might be infected, but it's not very efficient. And actually, what we then detected in brains, and with brains I mean like more than like 60, 70 that we checked by SARS positive single cells. So we do not see overwhelming amounts of virus in the brain. And what we also saw is that that does not really correlate with those tissues that show high activation of immune cells. So this might also because we are talking about old people, we are talking about people with underlying diseases and comorbidities. So what we saw might not even be due to COVID. So it's very broad pictures. However, other people also showed implicated neurons as a source for virus, or also the blood-brain barrier. So in another project, we also checked for immune or abundance of immune cells around the vessels in the brain, and we actually could see that there are several patients that showed immune activation and also recruitment of peripheral immune cells to perivascular spaces. So this led us to speculate that there might be something going on, at least in a subset of patients. And in another project, we also worked with peripheral vessels. So this is from the arteria carotis here, one of the main vessels in the body. And indeed, we also could show that SARS CoV-2 is abundant in a very small subset of endothelial cells, but it is abundant. And when we bulk sequenced this, we could also see that here case 4 and 5, in contrast to control 1 and 2, you see that there's quite some changes going on, and there were quite some genes downregulated in the peripheral vessels, and these were mainly in 4 categories. And interestingly, one category is the interferon gamma pathway, and this was downregulated in the peripheral vessels that we checked. But coming back to the brain, which ways are open for SARS-CoV-2 into the brain? I mean, we checked medulla in our first study and could not see so much virus positive cells there. So probably the medulla might not be the entry point for SARS-CoV-2. People checked nasal epithelium and the bulbus olfactorius. That might be one route of entry. But then we have the blood-brain barrier, and then if you have immune cell recruitment, you might also have vessel leakage. You might also have transfer of SARS-CoV-2 with the immune cells into the brain parenchyma, or you might just simply have blood-brain barrier dysfunction. And to sort this out in a bit more detail in an easy-to-go model, we at the University Medical Center in Hamburg teamed up with the Fraunhofer Institute, specifically with Ole Pless, who is the head of a biomarker and translational drug discovery, and they developed an iPSC derived human blood-brain barrier model. And this we actually used to infect cells in the transwell model. So the upper reservoir is then representing the vessel lumen, and we placed the SARS-CoV-2 from the vessel lumen, and the lower would then be represent the brain parenchyma, and the big question was, are these cells infectable at all? And then we did cytohistochemistry, and we could indeed see that a subset of cells get infected with SARS CoV-2. And when we quantified, we could see that this is about 0.5% of all the cells. So it's not highly efficient, but it's there. So we then checked for the receptors, and we could identify that ACE2, the main receptor, is low, but it's abundant on this brain cell, as are the cofactors TRPV2 or NRP1. And then we also checked in the available literature. And here, for example, for our endothelial cells on the blood-brain barrier, NRP1 is highly abundant and cathepsin L, but ACE is also abundant in traces like we detected in our blood-brain barrier model. But then we were like also doing immunohistochemistry on some of our brains, which is like really in line with the amount that we found in our iPSC model, but was what also like known from the literature. And but then we got kind of a bit stuck, and I was rather thrilled when I got from nanostring, because we like had to duck a bit or poke a bit like in the haystack, and we like checked some common dysregulation markers that were known in the brain. Here, for example, PKM1, but this was completely not like unchanged between control patients and COVID-19 patients. Thus, we thought, okay, we should really get a bit more specific on this. And as I said, we were rather like excited when I got the COVID grant, because it really offers to take these brains from these poor people that died from COVID and then really dissect only the areas of interest. And for this, I got the opportunity to submit 2 slides. And thus we checked, here are some of our paraffin blocks, and from the paraffin blocks, we specifically selected areas that did not so much differ between the immune phenotype, because they were actually more interested in the blood-brain barrier itself, like the endothelial cells and the astrocyte and feet, than to get like these rather strong immune phenotypes. As I said, we selected areas with like comparable amounts of astrogliosis or microglia activation. And from these, we then made like these like punches, and then we arranged the punches. So we always had like 6 different patients and control on one slide to get also like very like heterogeneous basics for settings for detection of our vessels. And so we in total investigated 5 control brains and 7 COVID-19 patients, and we submitted our slides to the R&D facility of NanoString in Seattle. And this was really, really very exciting, because then I could live select 48 vessels in the human brain, and for morphology markers, we choose CD31 as a like a vessel marker, GFAP for the astrocytes, so also to visualize the astrocyte and feet that are surrounding vessels, also again to exclude like specific vessels that have a super highly activated phenotype, and also CD45, which is the common marker for immune cells, and obviously DNA to show nuclei. And this is how it looked then. So our 12 brain punches, and here we choose cortex tissue because this is like center where you have cognition and where also things like dizziness and this brain fog thingy that you frequently have in COVID-19 or long COVID, like the virus you have. So we choose here the like frontal cortex, and here's an example of the vessels I picked. So we try to get like kind of a more or less like equal size, and I also try to excise them very thoroughly, so like only the vessels, and we picked like 3 to 5 vessels per brain. So I already told you a total of 48 vessels. And in this area, so we're like an average of about like 27 cells, but we had cell counts from 8 cells to 67 cells. So I was very curious whether like the smaller vessels with like 8 cells also gave decent results. And I was so happy because I really gave super good results, and the quality control turned out okay for all 48 reads. So when we then made kind of a heat map, and here the COVID-19 patients are in this green hue, whereas controls are in this like more like pink hue. You could see that already here in this overview, the patients or the vessel like transcriptome cluster really into groups that represent COVID patients very nicely. And when we then checked like for genes that are really highly significant, we could really sort out several genes that are upregulated in these endothelial cells in COVID-19 brains. We were very excited about this, and especially these genes that are upregulated, because some of those are also, and you might recall that we checked already peripheral vessels where the interferon gamma pathway is downregulated. And interestingly, and of note, interferon gamma pathway is upregulated in the COVID-19 brains. And I just saw another manuscript out last week where the interferon gamma pathway is also up in endothelial cells, and this was, we call it just bulk sequencing in the gut. So also another organ which is suffering from like COVID and infection, because you also have diarrhea and so on. It's not really sorted out what the SARS-CoV-2 infection is doing there, and especially 2 genes, EPTM1 and EPTM2, are really standing out in this analysis. So this is also ongoing research that we are currently doing to figure out what that might mean for patients with COVID. And so we are like a huge step further into elucidating what's going on in severe COVID-19. However, there are still a lot of questions to be addressed. And it's still, I mean, not highly clear what's going on in those brains, and so on, what's the long-term consequences of COVID-19. So probably there is, like in our hands, there's not so much going directly on with the virus in the brain, but there's going on a lot with vessel dysfunction in the brain. So this is really something that should be investigated further. And with this, I would like to thank my co-workers in the Institute of Neuropathology, where I'm located, mainly Christine Hartmann and Ella Thies, my direct co-workers and like collaborators in my work group, but also Ole Pless from Fraunhofer Institute here in Hamburg, other co-workers from the UKE and the NIMR, and which is Susanne Krasemann, because she did the virus infections of the cells, and Fabian Heinrich at the legal medicine, because he really gave access to super high quality like COVID-19 tissue, which was a prerequisite to get these super nice data with the nanostrength GeoMax settings. And I would also like again to thank the nanostrength people for giving me this grant, and also for Liang, Kati, and Jan to support me with my project. And with this, I'm giving back to Doug. Thank you a lot.
That's wonderful. I mean, what a presentation, Susanne, and you definitely made the most of accessing that technology. And what's just fantastic is your passion for this research coming across. So thank you very much for sharing that with us. We will come back to you, so don't hang up. You can disappear, but don't hang up. We'll come back to your questions at the end. Just a reminder to you all, can you please get your questions in, because we want to ask loads of questions of our speakers towards the end of this session. Before we come to our next speaker, we've got a little bit of audience participation. So if I could ask Alexis, who is in the background here, excellent, there is a poll. And so what we would like to hear from you all is around conducting spatial research. You know, what analyte will you be focusing on? Is it RNA? Is it proteins? Or is it both RNA and proteins? So if you can please click on this and your results will come in. We'll give you a couple of seconds. This is really useful data for the companies, and it will just be interesting to see how you want to access these technologies, I think. And so I think we'll close the poll there. And shall we view the result? I think I can click on this to view the results, so we can be open with the results. That's what we like to be. Interesting. So 0% just RNA. Good proportion of you, a third on proteins and 2/3 on RNA and proteins. So, well, thank you very much for filling out that poll. If we have time, there's another poll later on. We do like a little bit of audience participation here. Thank you for clicking on that poll. We now move on to our second talk, which will be our presenter, Dr. Michelle Naughton. Welcome, Michelle. And Michelle, as a bit of background, received a master's in neuropharmacology and supported research on the modulation of the glial scar before pursuing a PhD on the activation of endoplasmic reticulum stress signaling in myelination at the National University of Ireland, Galway. Her current project, which she is doing as a research fellow at Queen's University, involves studying the regenerative profile of immune cells in multiple sclerosis. So I believe the presentation, as it says here, will be to investigate tertiary lymphoid structures in MS. So Michelle, it is now time to hand over to you.
Great. Thank you so much, Doug. And yeah, no, it's been fantastic to be here and to hear about, you know, what that technology has already brought to Susanne. And we're a little bit earlier, we don't quite have, we haven't quite started yet with actually performing the profiling yet or in getting the data out, but we're so excited to see what it's going to be. So yeah, so just to explain, I'm from Queen's University Belfast in the Wellcome Wilson Institute for Experimental Medicine with Professor Fitzgerald's group. And in this group, we are trying to identify new strategies and new targets to for multiple sclerosis by uncovering new knowledge about how the immune system influences the central nervous system, whether that's by stimulating regeneration, but also looking at how it affects pathogenesis, obviously. And this project, investigating tertiary lymphoid structures in MS, we feel could have a big impact. And yeah, just to explain a bit more about who aren't as familiar about it, yeah, it's the most common disabling chronic neurological condition that affects young adults. So that's 2.8 million people globally. There's a strong female bias, 69% of them are female, and the average age of diagnosis is 32 years old. So that's just when people are forging their careers, their families, their relationships, and that even coming to that diagnosis can take quite a long time because it's a very heterogeneous disease. The range of symptoms that people can experience can vary from like numbness, tingling, to having difficulties with walking, bladder function, fatigue. There's a huge range of symptoms, and it can take quite a while to actually come to that diagnosis. But in the clinic, they are able to see common clinical patterns. So 85% of people will present with the relapsing remitting pattern of disease at onset, which means that their clinical symptoms will wax and wane. And then after a certain period of time, they enter what we call a progressive phase, where their disability increases in the absence of relapses. And for that other 15% of people with MS, they only ever experience that type of progressive phase of disease, and they would be called primary progressive. And the diagnostic hallmarks of MS are these white matter lesions that are detected by MRI in the brain that are disseminated in time and space. We know that when we look histologically, that these correlate to these areas of myelin loss, where here with myelin in blue, you can see that the very stark areas where the myelin has been stripped off the axons, that are now just completely white. You also see these paler areas here where there has been some repair that we call remyelination. And histologically, we do get like a lot more detail than you can do by MRI. And even going back to like 1916, they were able to see that the axons were still being spared, even while these phagocytic macrophages were stripping the myelin sheath that was around them. And even then, it was noted that even in the cortex, where there's far less myelin or next to none, as would have appeared to them then, that you could also see subtle changes in the cortex. But for a very long time, MS was considered a white matter disease. It was even considered to be a purely motor disease for a long time, and this caused untold stress basically for the people that were experiencing it until they really came to understand that it's a disease that affects the whole brain. And I think the history of MS research, it really goes to show how important a detailed spatial awareness of what exactly is happening in the tissue is so important to understand these processes and how it really has brought on our field so much. So I'm going to be highlighting a couple of these neglected pathological features that didn't receive attention for a long time, but then have made a real contribution to the MS field since we have done. And this is why I'm so excited about getting to use digital spatial profiling with our tissue, but we'll come to that. So the first one I was going to mention was the normal appearing white matter. So this is the white matter that, for all intents and purposes, like you would have seen here with the nice blue brain, you can see the very obvious areas that had been affected by the disease, but the other areas of blue are also under severe stress. And this was noted back in the 70s by our own Professor Jean Langhorn. And I mention her because she was a noted neuropathologist that established a very important tissue collection containing containing a whole range of cases of demyelinating and other diseases that is still really that's still capable of really bringing a lot of new knowledge to the field. So I wanted to mention that this was one of several seminal contributions that she made to the field, but she showed that 72% of samples of this normal appearing white matter were histologically abnormal when you actually look at it in detail. And this proved to be insightful because even just very recently, there's been a report that early tissue damage in the normal appearing white matter can characterize a subtype of disease, and that this has predictive value for disability progression and treatment response. So you can imagine about what difference that can make to, what difference this could potentially make for MS patients if you could actually gain that knowledge from knowing what's happening in the normal appearing white matter. Another example that was neglected for quite a long time was that MS is also a grey matter disease and not just a disease of white matter. Indeed, the lesion area in the grey matter can surpass the area of white matter lesions. It's as well as being an immune-mediated condition, it's also a neurodegenerative condition, and you'll see decreased neuronal density both in the normal-appearing grey matter and in the demyelinated grey matter lesion area in MS. And even more than that, we find that grey matter pathology is a better predictor of disease trajectory, that grey matter damage will correlate with disability milestones, in that if you have a greater extent of grey matter damage, that you tend to have an earlier onset of disease, earlier time that the person may use a wheelchair, and an earlier time to death, and that also that cortical atrophy of certain brain regions has been shown to be predictive of cognitive decline. So it's a really important aspect of the disease that we all that MS researchers are very conscious of now, that it's not just a disease of the white matter, that it's a disease of the whole brain, and if we want to, that we really need to understand the drivers behind grey matter pathology if we are to really tackle MS. So this brings us to like one more feature of the pathological feature of the disease, which I feel has been neglected for a long time up until relatively recently, which is meningeal inflammation, and this could be one of the key drivers of grey matter pathology. And this will be the focus of our project that we want to apply with using NanoString and Illumina's technology. So just to explain a little bit more about what we mean by the meninges, are these membranous layers that surround the brain and spinal cord. So there's three layers: the outer, more dura mater, then you have the arachnoid, which contains the subarachnoid space where you'll have cerebrospinal fluid, and then finally just a very thin membrane called the pia mater, which would be directly overlying the cortex. And but it not only just contains cerebrospinal fluid, it is home to numerous immune cells. And if you classify the degree of inflammation in MS patients, you'll find that those, if you compare between those with the least level of meningeal inflammation to those with the very highest levels of meningeal inflammation, that there is a very stark contrast in the level of grey matter pathology that's occurring just directly beneath them there, as you can see in these two examples. Much of the work that I'm going to describe here has been done by Dr. Owen Howell's lab, which is a very important collaborator of ours on this project. And one of the reasons why why I feel it's been neglected up until recently, which I meant to mention, was that these meninges were often just discarded for a long time after, during the brain dissection process in the anatomy lab, so they would simply have taken them off, which now just horrifies me because now that I know just how much information we can actually gain from them, and their own contribution that they can make to the disease course. So it's shown here with in this graph that having a higher degree of meningeal inflammation correlates to having, again, an earlier onset of the disease, being at an earlier age when you've reached that progressive phase, or reached the point in which you use the wheelchair, or indeed reach the point at which the patient dies. So it's incredibly important that we understand meningeal inflammation better. And as I said, that's what we're going to be aiming to do with this. This category of inflammation, the very highest category of inflammation, is where we see what I'm calling the tertiary lymphoid structures. And there's been a little bit of debate about them, but just to show you what our description of these lymphoid structures, you might also have heard of them as B cells, but they are semi-organized aggregates of immune cells containing T cells and proliferating B cells, amongst others, that are present and are usually found nestled within the meninges in the deep sulci of the brain, as you can see here by these red dots. Now, in this graph here, this wouldn't be a single MS patient, wouldn't contain this amount of follicles, but this is this is like a heat map, if you like, of the most common locations in the brain where you might find a tertiary lymphoid structure. So this is part of, and this work here that identified that tertiary lymphoid structures, although they're more common in secondary progressive MS, that they can also be observed in cases within even just a few short years before, few short years of disease duration before, unfortunately, the patient died. And one of this work was supported with tissue from the Dame Ingrid Allen tissue collection that we have here in Belfast, and so and that's, you can see that with some of the images here coming directly from this collection. So just to describe a little bit more about it, that it contains over 300 cases of MS and other neurological diseases. They're FFP tissue, and it ranges from going back to the 40s all the way up to the 1990s. So the vast majority of these samples are before the advent of MS treatment. And this means that you can look at the natural disease pathology and indeed the endogenous repair processes that occur in the tissue. And it also includes some very rare cases, including these instances of including these samples where they had a very short duration of MS before they died. So these would be relatively rare to find in tissue banks, and especially nowadays, thank goodness, thanks to the wide range of treatments that we have nowadays for MS. But these could be really highly valuable samples when it comes to finding out, what is happening early in the disease process. And that is what we want to do now that we are lucky enough to have access to this resource of tissue, and thanks to Dr. Owen Howe's expertise, that we'll get to investigate tertiary lymphoid structures in MS. And I'll just tell you a little bit more about that now. And so I am working with, I'm co-supervisor for Dr. Rachel Key, who is a PhD student on this project, and she is supervised between Dr. Owen Howe's lab and the lab here with Professor James Fitzgerald in Queen. And she has been doing a lot of work with updating the clinical classifications. So as you can imagine, with these cases, some of them dating back to the 1940s, so we need to make sure that these are clinically up to date, that they correspond to modern classifications for MS, and that we can definitely send over that there is sufficient clinical information with them that we can properly have them defined, so that we can shortlist them properly for use in these studies. And we've been conducting comprehensive profile of inflammatory indicators in order to conduct this tertiary lymphoid screening. And this is all with the aim that we can provide the first comprehensive profile of the cellular composition, as well as the transcriptome of tertiary lymphoid structures, which I feel is going to be a major contribution for this field. And we'll be doing this via multiplexing and histochemistry, as well as the genomics digital spatial profiling with nanostring and Illumina, which we're going to be so excited to see. And we're also hoping then that because with the collection, we'll be able to compare changes in these structures between those with a short duration of MS versus those with a long duration of MS, so that we
Hello, everyone. I think we may have lost Michelle just at the end of her presentation. We have had a couple of technical issues this afternoon, but I can assure you it was just before the last slide, so I'm pretty sure she presented most of her research. Hopefully we'll get Michelle joining us again before we end the webinar in a few minutes' time, so we can ask her a couple of questions. But until then, Susanne, can we have you return to the digital floor and begin the Q&A session? Yes, we have Susanne. Hopefully Michelle will join us in a second. So we have had some questions in, which is fantastic. People, if you still have some questions, there's time to put them in the box. They're flashing up on my screen, and I will ask them of the speakers. So, Susanne, I wonder whether to start off, and again, fantastic talk. Could you tell us a little bit about whether you've detected the virus in neurons or astrocytes, and whether you investigated other brain regions as well? And is there an abundance of SARS CoV-2? Does that change across the brain?
So in the first study that I also presented today, we focused on medulla because this was one of the putative entry sites, like going like up the spine and enter the brain, while there were like nerves that like goes through the or into the medulla, also coming from the lungs or so. And we were really surprised to not see like really much virus. There were like about 50 cases positive for RNA. However, 50% of cases positive for RNA. But actually, we never saw SARS CoV-2 in like specific brain cells. I have some unpublished data where I tried to do, or where I did like triple fluorescence staining, and there it still looks as if it's rather immune cells that carry the virus into the brain. However, you also have to be cautious because we are always talking about diseased people, and even in the lung you might not find virus because it might have been cleared in the meantime.
Great, thank you, Susanne, and welcome back, Michelle. We just lost you at the end of your presentation there, didn't we?
Yeah, but I think you got the gist of it.
Great, and I mean, thank you for your presentation. It was fantastic. Even though we lost the end of it, we managed to follow it all, and actually, it's very close to my heart because of my time of working at the UK Multiple Sclerosis Society. Very much keeping an eye on the field and how that's developing. So, thank you very much for your insights. So, we just asked a question of Susanne, and actually, I'm going to ask you a follow-up question because you mentioned the lungs and versus the brain. Did you see that the abundance in the brain correlated with the abundance of virus in the lung?
No, not necessarily. So I mean, the abundance is very rare. So what we see is that mainly, and this is also a problem when you take bulk tissue for detecting SARS with RNA techniques. It's more correlating with viremia in the bloodstream. So this is also something you have to be careful about, because if you have highly tissues with a high blood flow, you might just detect virus that is still abundant in the blood. So I mean, as I said, we only see some of these cells still left in the brain, but it does not correlate.
Okay, no, that's really fascinating. And Michelle, coming to you, these tertiary lymphoid structures, you know, how common are they in people with MS, and is it specific to MS or other autoimmune conditions?
Well, it's been found in up to 40% of SBMS cases looked at so far. In terms of short duration or relapsing remitting MS cases, there haven't been enough looked at, but we'll be hoping to contribute with that information using the collection that we have here. So once we have that screening finally completed, we'll be able to conditions. It does. Sorry, I hope my connection is still working. Yeah, it is found in other autoimmune conditions, and it's usually associated with a more severe disease. It's also found in certain tumours and cancers, where it's actually been found to be beneficial in some cases with helping to resolve those tumours. So yeah, we can definitely learn from those other conditions as well.
Okay, no, that's fantastic. And I mean, what's the significance, do you think, of this? And there's still a lot to discover, but for people with MS who are living with the condition, and you know, is this something that can help sort of diagnostic, prognostic, treatment, other?
Yeah, no, that's definitely what I hope we'll be able to find with this research, because those that have this high degree of meningeal inflammation, which appears to be driven by these tertiary lymphoid structures, that they're driving this cortical pathology, that they're having the most severe cases of disease. So, they're having a greater disease burden as a result. So the more that we can understand about these, I'm hoping that we'll be able to find novel strategy to help to counteract it.
No, that's great. And a similar question to you, Susanne, and I think we should probably make this the final question, given that we've reached the end of the hour. But the significance of your research findings to COVID-19 patients, or any of us that could be affected by COVID-19, how does this these insights impact on maybe understanding the disease, and again maybe treating, preventing the disease in the future?
Naja, it's a question. So there are the question would be to avoid contact of SARS CoV-2 with brain cells. So like you could act on the very beginning, because now also with diagnosing or detecting SARS CoV-2 infections much earlier, you could use inhibitors. But probably it would also be good to really target the immune system and this interferon gamma responses. However, we did not do research specifically on this yet. So this would probably also be something that you could first start to evaluate on the blood-brain barrier model. Because, and this is also one point, I mean, we always have end stage when we investigate paraffin-embedded tissues. So it's also something that would be worth studying in the people that recovered from COVID, that you follow up if they have really also interferon gamma signaling problems, if there is a dysregulation going on and such.
Great. That's wonderful. I mean, I could ask questions of you all evening long, but I'm sure you've got lots of things to do, and people viewing have lots of things to do as well. So I'm afraid we're going to have to end the questions there. Apologies for some of the technical issues. It wouldn't be a webinar unless there were some. So I'm sure you have every sympathy for us. And I just wanted to say a huge thank you to the speakers, in particular Susanne and Michelle, as our award winners. Clearly, you have and you are making a huge amount out of winning this award and having access to this technology, and look forward to seeing how your research develops. Also want to say a big thank you to the companies. We heard from Agnes and Pavel earlier. NanoString, Illumina have been fantastic to work with, and it's been a real pleasure. And we at the British Society for Immunology look forward to working with them again in the future, perhaps around another award scheme, but plenty of other options, I'm sure. And I'd like to also thank you as the audience for joining us this afternoon, this evening, this morning, wherever you have joined us from around the world. I saw a very international audience when I had a click through the participants there. So thank you very much for giving up your time, and I hope you enjoyed these wonderful presentations from our award winners. I'm told that I need to remind you that this is number 2 in a trilogy of webinars, and all the best things coming threes, of course, don't they? And the third and final one is tomorrow, which is a focus on neuroscience. So if you haven't already and you are interested, please do sign up for that final webinar tomorrow. So I think we're going to close the webinar with a short video from the companies. So please do stay on and watch that. But that's it from us. Again, thank you to the speakers. Thank you to the organisers. It's been a pleasure chairing you this afternoon. Do take care, and goodbye for now.
After completing slide preparation, which is similar to IHC or ISH, we will put our slides into the tray, and then we'll put the tray into the instrument and load buffer. Next, we will be ready to enter our slide records and scan parameters. A fluorescent image with up to 4 colors is generated to enable selection of region of interest or ROI on this tissue. You as the user can choose these ROIs from anywhere on your laptop. Geometric profiling allows us to choose circles, squares, or even polygons. This would be a great tool when you have pretty homogeneous regions like immune cells and the tumor segregated within one tissue or across tissues in your cohort. Geometric profiling was utilized in David Rimm's Nature Communications article in 2020. Many users can also create rules for segmenting the ROIs. This would let morphology of the tissue determine the boundaries of the area of illuminations, or AOIs, to enable 100% cellularity. For example, users can create tumor and TME segments across a tissue sample as shown on this example. Selected ROIs are illuminated with UV light to release barcodes from DSP probes. And then these DSP probes are deposited into a 96-well plate. The released DSP barcodes are prepared via a standard library preparation, so they can be read out on Illumina sequencers. The data can be mapped spatially back to the originating region of interest. Samples can now be loaded onto your Illumina sequencer. Depending on your experimental design and the instrument capacity, you can read up to 760 ROIs from a single geomics experiment in a single Illumina run. Here we have an XE 2000, which will allow us to easily read hundreds of protein or RNA analytes. Once the FASTQ files are generated, they'll be converted to digital code counts and then uploaded back to Geomics DSP instrument. The data analysis screen is divided into 3 panes and a taskbar. Here we describe the functions of each pane and the interactions between all 3. The scan pane is shown on the left side and contains individual viewers of the scans. In the middle pane, we have our datasets and probe lists. On the right, we have our visualization pane, which shows the various graphs. In this case, this is our QC graph. The middle datasets pane lists all datasets and probe groups associated with the current study. The visualizations pane is on the right. Visualizations are visual representations of the dataset selected and probes selected, and the adjustments applied to the dataset from those probes. Now I'm going to open another study. Here we can see the data has been QC'd and normalized. Once you normalize the data, you can see in your visualizations pane different graphs, including pathway maps, pathway tables, bar graphs, correlation plots, box plots, PCA plots, trend lines, and we can view heat maps. I'm going to end with clicking on a data point on the data visualizations pane, and you'll notice that ROI associated with it will be zoomed in on the left pane of the screen, presenting a seamless integration of the images and the digital counts. Thanks for joining our Customer Experience Lab sessions.